Well, hey, welcome back. This is your next section of notes over the neuron. This is our second section of notes in the nervous system. Remember, as we go through the slides together, the headings are the things that are in black, and the vocabulary terms are in red, and important details that you need to know for future assessments are in green. All right, get your notebook out, write this down in the input side, which is the right-hand side of your notebook. And here's your essential question to be thinking about as we go through these notes. List the things that are moved via axonal transport. As we go through the notes, we're going to be learning about this and discussing this. So pay attention as you go through there and go back and answer this question. Also, guys, remember, we are taking these notes in Cornell note style. So when you're done with the notes, go back and summarize them and add some questions in the margin that you think might be test-worthy questions. All right, this picture, this image right here is a microscopic image of a neuron. We can see here the cell body with the nucleolus right there in the center, the nucleus and the nucleolus. And then we've got the extensions, which are the dendrites and the axon, okay? All right, the dendrites we're going to discuss first. The dendrites are kind of these tree branch looking things coming off the cell body of the neuron. And these conduct impulses towards the cell body. So the dendrites are receiving information from another source, maybe another axon or other cells that are involved in the central nervous system. They're receiving the impulses and they're bringing the impulses towards the cell body, so towards the nucleus. The dendrites themselves are typically short, they're highly branched, and this is important, they are unmyelinated, okay? They don't have any myelin around them. Now, the surfaces of the dendrites are specialized for contact with other neurons. So these extensions here are specialized to make contact with other neurons. And the dendrites themselves can contain neurofibrils and Nissl bodies. Okay, so the neurofibrils are these little things, and the Nissl bodies are these little things. It's kind of hard to see in the picture right there. Now the axons, some neurons can have a couple of axons. Most of them just have one. What it is, it's a long, thin, cylindrical process of the cell. It arises from an axon hillock, and the impulses arise from initial segment, which is the trigger zone. All right, the trigger zone is right around here near the axon hillock. And there could be side branches of axons, which are called collaterals. And the fine processes down along here that make contact with maybe a muscle cell or other neuron would be the axon terminals. The swollen tips at the end of the axon terminals are called the synaptic end bulbs. The synaptic end bulbs, if you remember, are the things that contain the vesicles filled with neurotransmitters. And when we studied muscles last unit, it was the synaptic end bulb that came into the neuromuscular junction. And you can see here, if you pay attention to all of this information over here regarding the axon, there's a lot of stuff. There's a lot of anatomy involved with the axon of the neuron. Okay, now axonal transport. This is really important. The cell body is the location for most of the protein synthesis, okay? So remember, protein synthesis, what is protein synthesis? It's a process of getting the DNA, copying the DNA, transforming the DNA into RNA, and then the ribosomes meet up with those amino acids and it makes the new proteins, okay? So, because nervous tissue cannot regenerate. It does not go through mitosis. The nuclei can't reproduce themselves. But what can happen is it can make all kinds of new proteins and stuff like that for neurotransmitters and repairs. Remember, neurotransmitters are those chemicals that are released at the axon terminal in the synaptic end bulbs to transmit the action potentials across. All right, the axonal transport system moves mitochondria, lipids, synaptic vesicles, proteins, and other cell parts to and from a neuron cell body through the cytoplasm of its axon. So we can see here, this is the cell body down in this picture, and the axon is the long cylindrical portion. And this is how transport goes from cell to cell in the nervous system, and also from the end of the axon back into the cell body. So transport in the axon can go in more than one direction. It can go away from the cell body and to the cell body, just depending upon what is happening. All right. Slow axonal flow, which is protein movement, that'll move one direction only, and this moves away from the cell body. As the proteins are produced in the cell body, then they move away from the cell body to the axon terminals. And they move about 1 to 5 millimeters a day. Okay, that's slow. And fast axonal flow, it moves organelles and materials along the surface microtubules. Okay, so on the surface of the axon, these things move very quickly, and they move 200 to 400 millimeters per day, which is not a huge distance overall, but when we're talking cells, this is huge. This is fast, and this is quick, and very long distance in terms of a cell. Fast axonal flow can go in either direction. It can go into the cell body or away from the cell body. And this is really good for recycling inside the cell body, and also for those repairs that need to be done on a regular basis for homeostasis. Okay, so we've got two key terms here, anterograde versus retrograde axonal transport. Anterograde is going away from the cell body out to the axon terminals. Retrograde is coming back to the cell body. All right, we can see the cell body where the Golgi apparatus, remember that's the packaging system, that packages up those vesicles for things to be transported through the axon. That can be the neurotransmitters, other proteins, other things as well. All right. Also, inside the nucleus, we have the nucleolus, and other stuff can be coming into that region for processing. Okay, so neurons have many classifications. We're going to focus upon these ones. The sensory neurons, which are also called the afferent neurons. These are responsible for your transport of sensory information from your skin, muscles, joints, your sense organs, and your viscera to your central nervous system. Okay, so what are they doing? They are receiving information and bringing it from your sensory organs to your brain and spinal cord, your central nervous system. All right. Next classification is your efferent or motor neurons. These guys send motor nerve impulses to your muscles and glands from your central nervous system. Okay. And then your interneurons, which are called association neurons, these are in between the sensory and motor neurons. So they connect sensory to motor neurons together. And guys, believe it or not, 90% of your neurons in your body make up interneurons. Now for structural classification of neurons, this is going to be very useful for your little activity for today. You're going to be drawing some examples of these different types of neurons and coloring them in and labeling them. We first have these different types of neurons. They're based upon the number of processes found on the cell body. So for example, we got the multipolar neuron, which has several dendrites and one axon. So multipolar, we've got all these dendrites branching off, and then there's one axon coming off. All right? This is the most common type of neuron that is in the human body. Bipolar neurons have one main dendrite and one axon. All right? These are for your special senses, such as your retina, the back of your eye, the inner ear, and your olfactory, which is your smell, your sense of smell. Bipolar neurons are found in those special senses. Lastly, here's the unipolar neurons. There's one process only, and it develops from a bipolar, and they're always in your sensory neurons. All right, so unipolar means that there's only one process coming off of the cell body, and then it branches into the axon and dendrites. Okay. All right, so here's another image of that same microscopic view of the neuron. These little dots all around the outside that kind of help us with connective tissue and so on are the neuroglial cells. And the next time we take notes, we're going to talk about those in great detail. Here we've got the nucleus with the nucleolus in there. The cell body is a large portion. Axons or dendrites are located here. It's kind of hard to tell with this view that we've got right here. Okay, so this is kind of an introduction into our next section of notes, the neuroglial cells. Neuroglial cells are really, really important for the functionation of the nervous system, and they take up about half of the volume of the central nervous system. They're smaller cells than neurons, and they're about 50 times more numerous. Now these glial cells can divide, all right? And oftentimes the rapid mitosis of tumor formation or gliomas is the problem that people have when they develop brain tumors or tumors on their spinal cord. It's these cells right here that are dividing uncontrollably. There are 4 types of neuroglial cells in the central nervous system: the astrocytes, the oligodendrocytes, the microglial cells, and the ependymal cells. And you can kind of see a little illustration of each of those. And then the 2 types of neuroglial cells that are in the peripheral nervous system are the Schwann cells and the satellite cells. All right. Next time we'll be taking notes in more detail over the neuroglia and some more information about that. But hey, stop there. Go back and answer that essential question from the very beginning of the notes. Make sure you read through what you wrote, think about it, and come up with a good summary. And also highlight those key terms and come up with a few questions in the margin as to what you think might be test-worthy material. All right, I'll see you in class as we work on drawing and coloring and labeling the various types of neurons. Have a great day.